And right now, though, I have a special guest with me who will be talking about designing with biology, Dr. Pamela Silver from the Wyss Institute at Harvard University. Please join me in welcoming her today.
Hi, good afternoon. Thank you all for coming out. It's great to see so many people excited about biology, and especially designing biology. And I'm particularly excited to see about half women here, so that's exciting also. So I'm going to give you a brief story about what we're doing in terms of designing biology for what we think are real-world applications. So hence my title, Designing Biology for a Healthy World. So let me begin with lava. This is lava. And if any of you have ever visited, for example, a volcanic island like in the Galapagos, this is what they start out looking like. This is a brand new island. There's nothing there. And then after not so long, they start to look like this. So this is the result of birds flying by, dropping seeds, evolution in action. So this is what the power of biology. It can go from nothing to this beautiful, lush green landscape. So this should give you a feeling, if you don't have one, for the power of biology. So we like to think that we can use biology to solve some of the problems facing the world. And in that context, I think that biology is the most important technology of this century. So if silicon was the technology of last century, biology is the technology of this century. So I'm going to give you some justification for why I say that. So first of all, let's think about what biology does compared to what you might know about engineering. So biology is exquisitely sensitive. Your nose is capable of sensing a single molecule of an odorant. Now, I often ask people to guess whose nose that is. That's a test for afterwards. I'll give you some hints. It's female, it's no one in the audience, and it's someone pretty famous. All right. Biology can also send and receive signals very rapidly and efficiently, very specifically. So, of course, in your nervous system, you can sense odors and they register in your brain, but also single cells, bacterial cells that live in your gut can sense signals very rapidly and respond. Biology, like tools or like different kinds, the classic example is a rifle that at the beginning of the Industrial Revolution was shown you could take it apart and put it back together so you could sell it as parts. That was revolutionary. So that's the case with tools. We also know that's the case with biology. So biology, like engineering, is highly modular. We can take parts and put them together to make functional systems. And lastly, something machines can't do, at least now, is self-replicate. So biology, of course, like all the cells in your body, cells in plants, can duplicate themselves by dividing. And so imagine if you have a biological device, then that device, that cell, is capable of regenerating itself. So it's like a machine that can grow. And lastly, biology is exquisite at harvesting sunlight. Sunlight, I'll talk about this in a minute, is one of our greatest natural resources, and biology knows how to harvest it and do things with it. So there are a lot of things that biology can make. As I mentioned, it can make a leaf. It can make an organ. On the right there is spinning spider silk, which is one of the best materials there is. These are just a few examples of the kinds of things we would like to do with biology. Now, how does biology work? So I've shown here a bacterial cell. This is one of the simplest cells which we understand in detail. So that's a bacteria. It's like one that might live on your skin, lives in your gut, lives in the soil. It's a single cell. It has a tail that helps it swim, and in the middle it has DNA. So if we think of this in parallel to a computer, it has compartments, it can store information, and it's a chemical factory. So we think of the bacterial cell in parallel with a computer. Ideally, we would like to use the information of biology to build biological computers. So if we really reduce the biology of life to the simplest, to what it's composed of, there's DNA, which is sitting here. Well, this is a model of DNA. DNA is sitting everywhere. The DNA encodes the information, and we can edit that code. And then it produces RNA and proteins, which then articulate the code, and the cells do something. This is what's called the central dogma. It's the basis of all life. There are some exceptions. It's sort of like the English language. There are always exceptions, but this is the fundamental basis under which you program life. So if we're programming life, we want to do it simply and elegantly. So as Steve Jobs said, if we're designing biology, it's really about the customer's experience. We want, for example, a cell that can produce on command, but we want it to be simple and elegant. That is, it might be really complicated to me, but if you're using it, I don't want it to be complicated. So much like these computers, to be honest, I don't have a clue how they work, but I can sure sit here and type and send email and edit this talk, right? So that's how I want the cell to be. Now the question is, do we know enough about nature to actually do this? And that is really the essence of synthetic biology, is testing the idea of whether we know enough to actually produce something that's simple and elegant. Now we are audacious enough to think that we can, otherwise we wouldn't be here. But I think it's a work in progress. And it's learning, in essence, how to make cells and ultimately organisms or organs that we can engineer in a predictable, fast, and cheap way. And that really was the underpinnings of synthetic biology when we articulated it back about 15 years ago here at MIT. So these are some of the things that we're doing in my group at Harvard and at the Wyss Institute. We would like to address real-world problems by designing biology. So we'd like to convert sunlight to useful stuff. We'd like to invent new therapeutics, and we would ultimately like to transform the economy. So for example, and this is something that there's a lot of interest in Washington, D.C., and funding, is to get away from an economy that relies on petroleum. So everything around you is based on chemicals, your clothing, these chairs, and chemicals typically are derived from petroleum. So of course we would like to get away from that because for economic reasons, for self-sustainability reasons, and also ultimately if we can go directly from sunlight to chemicals, that would be a greener life. So we can imagine that biology is really good at chemistry. So we could use cells to do chemistry, to make materials, to even make materials that you've never seen before. We could imagine that we can make biology make inorganic substances that would be used in computer chips or sensors. And we can make new drugs. And ultimately, it would be fast and cheap. So this is the vision for synthetic biology in the future. Now about solar. So the whole idea here is that we would ultimately use the power of sunlight in some way to convert that energy into productive molecules. So let's think about sunlight. This picture of the Earth here illustrates what's called the Sun Belt countries. Depending on if you're from South America or Africa, you can see Australia is one of the Sun Belt countries. Obviously Brazil, where sugarcane is grown. Some of you may know that that Brazil is the pioneer in an ethanol-based economy for fuel, and that's because they are the world's biggest grower of sugarcane. In fact, Brazil anticipates doubling their sugarcane crop, barring that they don't go back to oil because of the recent oil strike. But if they double their sugarcane crop, they will be the global supplier of ethanol. Sort of puts corn into perspective. So this is the amount of solar power that hits the Earth, and we only use a very small fraction of that. The TW stands for terawatts, which is a unit of energy. So we would like to use what's hitting the Earth in a more efficient way. Now, plants, of course, are the traditional way that we think about harvesting sunlight. The curious thing about plants is, first of all, they were one of the first things to evolve, so they evolved billions of years ago, and they have a process called photosynthesis, which we understand in a fair amount of detail, by which they can convert sunlight to energy. You all learn about that, right, in grade school, right? So photosynthesis, the process of harvesting sunlight, actually evolved 3.5 billion years ago or so. It evolved when the atmosphere essentially had no oxygen and was high in CO2. So the process of photosynthesis formed when there was very little oxygen around. Once the Earth became, and for example, when the asteroid hit the Earth, and I think I have the timing wrong on this, and at the Science Museum that's particularly embarrassing, but it killed all the dinosaurs. So that was very sad, but it also caused the CO2 levels to drop rapidly. And these plants were sort of stuck because they didn't have a need really to evolve in this changed atmosphere. So this makes the process of photosynthesis relatively inefficient. So we would like a system, one of the things we'd like to do in engineering biology is make the harvesting of light more efficient. And there are many ways to think about this, and I will challenge down there, if you want to pick a topic, what you should do is make a more efficient plant. That would be revolutionary. What we have decided to do as synthetic biologists is combine chemistry with biology to make what we call the Bionic Leaf. We did this together with chemists, so this is a synergy between biology and chemistry, where the chemists developed ways to very efficiently produce hydrogen from water. This is in essence what photosynthesis does. It does what's called the water splitting reaction. And what we did was to create a catalyst where, in response to sunlight, hydrogen would be produced. So that's the sort of upfront part of the Bionic Leaf, but what do we do with that? And one of the challenges of harvesting solar energy is how do you store it? And how better to store it than in a biological system? So there are bacteria that are capable of eating hydrogen. They can live on hydrogen and carbon dioxide. Think about that. There are a lot of weird bacteria out there, and we need to study them and capitalize on them. And this is a case where we've done that. Furthermore, the bacteria not only eat the hydrogen, they make stuff. In this case, we can have them make a simple biofuel. We can have them produce commodities. And so in this system, which is what's called a drop-in system, it's cheap, and we can produce different kinds of chemicals. So this is an example of the artificial leaf. This is the inorganic part of it producing hydrogen. And then we interface it with a bacteria that's called Ralstonia that, as I said, can eat hydrogen and CO2 and make things like plastics, sugars, and fuels. It's pretty cool, huh? And so here's the whole integrated system. And this is what it looks like in my laboratory. Pretty simple. It's essentially a jar with some electrodes coming in. But the interesting thing about this, and you can go home, if you take nothing else from this, you should remember this, that we can make the solar to biomass conversion upwards of 5%. Now that may not sound like a lot to you, but the gold standard in biofuels research is algae. And algae it operates at about 5% efficiency. So in an artificial synthetic system, we've made a bionic leaf that's comparable to algae. It's drop-in, it's cheap, it can be scaled up, and so if nothing else, we envision a situation where this could be used perhaps locally in the developing world, also perhaps scaled up, but Dan Nocera and I, who's my collaborator at Harvard, have this vision of using it as a localized production. So think about the idea of personalized production. I know that I'm asking you to suspend belief for a minute, but imagine if you could have something where you could make whatever you wanted. You could program the cells to make a drug of your choice, a vitamin, a food. This takes away the power from industry and allows you to do it locally, which is something to think about. All right, so let me just finish by talking about biological computing. So we would like a cell that can sense an event, count forward, and do something. What it does, it could make color, it could change itself, it could make a chemical, as I illustrated before. It could make a therapeutic. It could die. We know how to get cells to do all of these things. So we can do each one of these parts, and ideally we would like to put them together and make a real biological computer. All of these examples have in common that cells will sense something and do something. Now, those cells could be the bacteria that I began with. They could be a yeast cell, which I see there's some sitting right here, or it could be an animal. Or it could be a cell living inside an animal, a cell inside your gut, or in the gut of an animal. So we engineered a cell that, and I want to be clear, no humans were used in this, with the possible exception of graduate students who made the cells. But I think this, if nothing, we've done experiments in animals where we can feed a bacterial cell to an animal, and that bacterial cell will behave like your gut bacteria. And it will go through the animal and report on what the animal has been exposed to. So for example, if the animal's been exposed to a drug, it will come out colored in sensing that animal has been exposed. So what's this good for? So as I said, it could be a diagnosis, positive or negative, for whether, for example, an animal has inflammation, has been exposed to a pathogen, or has been exposed to a drug or an antibiotic, for example. Secondly, these could also act as engineered probiotics. So once in the gut, they could hone in on a certain part of your gut, for example, a site of inflammation, and secrete something that would neutralize that inflammation. So that would be an example of a living therapeutic. Why do we want that? Well, there's a lot of problems that are chronic in the population. For example, a lot of inflammatory bowel disease, Crohn's disease. This gives you an example of a healthy gut in the top picture and a diseased gut in the bottom. For example, enteric dysfunction is a huge global problem, and it actually affects the IQ of children as they develop. And there are many diseases like this, and they are chronic diseases that cause people discomfort and pain, even in the first world, and predominantly in the first world in some cases. And so you could imagine these being actual probiotics that people might want to use. So as I said, we imagine a cell which we can engineer quite readily that would act as a diagnostic or a therapeutic. And let me just give you one example. So the green cells, you're going to watch the cells grow here, and they're growing together, and one of the cells is going to start secreting something that's going to stop the growth of the green cells. So you can see the dark cells taking over and the green cells stop growing. And that's an example of cells secreting a drug that can kill the other bacteria. So we can imagine that we could do something like this in the gut. So I'd like to present the idea that synthetic probiotics, not only could they be used in humans, but they could also be used in livestock. I've been interacting with the livestock industry. I never thought I'd be talking to chicken farmers, but it turns out in chickens, inflammation is a huge problem. And in the context of livestock, where the population is very isolated, you could even imagine using synthetic organisms. They would be contained. I want to say one last thing about using synthetic organisms that I think is going to be transformative. Now, some of you, and well, you should be, worried about what happens when these organisms get out into nature. And that's a big issue. in synthetic biologies. Of course, in many cases they will be contained, but if some of these examples I've told you about come true, there would be a case for organisms being allowed into nature. Now, we have a way to get around that, and it's going to be revolutionary in the next 5 years. And that is the ability to resynthesize entire genomes of cells. Part of the gestalt of synthetic biology is that it will be faster and cheaper. And one of the underpinnings of that is the ability to synthesize DNA that would encode an entire organism. This is something we're aggressively working on now, and I believe this will come to pass in the next 5 years. And that will become the gold standard for industry and anyone who is making engineered organisms. And those organisms will be safe and unable to undergo recombination with organisms out in nature. So I want to leave you with that thought, and then lastly, tell you about one last project. And I noticed that Neri Oxman has a display right up there, so you can go see her work. We're working together with her in the Media Lab because she's interested. She's a designer. She's actually designed 3D printed clothing that were on the Paris runway. But she met one of my students and got interested in the idea of integrating the clothing with integrating the clothing with bacteria that can harvest sunlight. And together we could make photosynthetic clothes. And so I invite you to go, this is a project called Mashtari. There's a TED Talk on it, and I invite you to go look at that, and you can see what we're doing, and they're going to be on display at the next Olympics. So this is an integration of design and synthetic biology. And I'll just conclude with, I hope I gave you some ideas and ask you, what do you want to build? And then you can come work in my lab. So thank you. Thank you.